Hi, everyone, and welcome to this week's edition of the Extracellular Vesicle Club. So the paper that we're going to be hearing about today is very near and dear to my heart because I've been collaborating with Matthias Ostrowski in his lab for some time, and I had the privilege to host Martina Fabiano in my lab for about a year as a visiting student. So today, Martina and Alan, her co-first author, are both in Europe. So they've moved from Argentina to Europe on new adventures in EVs. And I'm excited to welcome them here today to present their recent work that was just published in Cell Reports. So as many of you know, the reigning paradigm of EV-mediated communication over the last couple of decades has been that EVs travel between cells and they transfer their cargo to other cells. But there's another way that EVs can also be functioning, maybe several other ways that EVs can be functioning that have been known for some time, but that I think there's more and more evidence that's coming out that shows that EVs can also have other mechanisms of action. And of course, this does not negate the fact that EVs can transfer cargo molecules as well. But surface activity of the EV is something that is increasingly coming to the fore in the field, also with studies of the so-called EV biomolecular corona. And so today we're going to be hearing about some modes of surface activity of EVs. I would just like to ask everybody who's on the call to please put your comments and questions in the chat box. And then after the presentation, we will call on you. And if you'd like to, you can unmute yourselves and show your video then. So let's see here. Alan, Martina, I'm going to hand this over to you, and we look forward to your presentation.
Thank you very much.
Okay. Thank you for the introduction. We start sharing. Okay, so what we want to show you today is the data that we recently published as for our latest work together with Martina, and it's titled Plasma Extracellular Vesicles Surface Located Gas-6 PROS1 and CD39 and CD73 attenuate inflammation. This work was mostly made in Inverse Institute in Argentina and with the collaboration with Hopkins for the Witwer lab. So I will put the laser and we'll start. So I'm going to talk a little bit about the acute inflammatory response. So the inflammation is a defense mechanism that promotes the clearance of harmful agents, including invading microorganisms, and is initiated through coordinated vascular responses that enable the rapid influx of leukocytes and plasma factors into the affected tissue. So this response is coordinated by several families of pro-inflammatory factors, including cytokines, also chemokines and lipid mediators such as leukotrienes and prostaglandins. Of course, macrophages, whether tissue resident or derived from circulating monocytes, are highly adaptable cells that orchestrate the initiation of inflammation. So upon pathogen recognition, macrophages rapidly mount an inflammatory program characterized by the production of pro-inflammatory mediators, including cytokines such as IL-6, TNF, and IL-1 beta, among others. And macrophages exhibiting this antimicrobial, highly inflammatory profile are commonly referred to as inflammatory macrophages, or classically activated or M1 macrophages. Of course, inflammation is necessary and beneficial, but it needs to be tightly regulated in order to avoid tissue damage and chronic inflammation. So, the activity of these macrophages needs to be temporarily controlled to avoid the perpetuation of the inflammatory response. So during this resolution of inflammation, macrophages shift toward an effector state, characterized by anti-inflammatory and pro-repair activities, including the secretion of the cytokine IL-10 and the specialized pro-resolving lipid mediators such as lipoxins, myosin, and resolvins. So this phenotype commonly referred to as resolutive macrophages or M2-like macrophages, supports tissue regeneration and restoration of the homeostasis. So the resolution of inflammation is critically dependent on cytosis, a macrophage-driven mechanism responsible for the removal of apoptotic cells, mainly granulocytes accumulated as sites of inflammation. So the interaction of between macrophage and either live or dead neutrophils is a trigger for these resolutive mechanisms with a strong downstream anti-inflammatory signaling. And this is after the recognition of the soluble proteins, Gas6 and protein S, which bind the phosphatidylserine exposing apoptotic cells. Then they are recognized by the TAM family of receptors, which includes TYRO3, AXL, and MERTK, expressed by, for example, macrophages or dendritic cells.
So I am going to go a little bit back on the inflammatory response. As Alan said just now, all the inflammatory response, it's very important for like removing all pathogens and restoring homeostasis. And one of the most important cells and key cells that infiltrate the inflammatory tissue are neutrophils. They are specialized cells which are key for phagocytosing and destroying, for example, bacteria and fungi. And they do that by, for example, a myriad of different functional effector functions, such as the granulation, that is the fusion of a wide array of granules with proteases and enzymes that are damaging different pathogens, but also, for example, with their oxidative burst, they secrete a lot of reactive oxygen species within the phagosome or to the outside of the cells. And they also secrete different cytokines, for example, IL-1 beta, and this also needs to be tightly regulated because otherwise it will lead to tissue damage, as Alan said before. For that, one of the mechanisms that neutrophils have to regulate or to stop inflammation is that they are really short lifespan cells, which means that they soon after they infiltrate the inflammatory tissue, they die by apoptosis, which enables the phosphatidylserine exposure on the membrane. And then what Alan told you, that macrophages are able to phagocytose them in a process called efferocytosis, which leads to this phenotype change in macrophages. However, we started thinking about plasma extracellular vesicles being able to infiltrate the tissue because of the changes in the vasculature and achieving higher concentrations in the tissue. So we started asking ourselves if those plasma extracellular vesicles were able to interact with the different cells, such as macrophages and neutrophils, and modulate their responses to the different pumps, for example, viral pumps or bacterial pumps. So that was our hypothesis. So after that, we started studying a little bit more about this.
So we previously showed that EVs attenuate macrophage inflammatory responses. So when macrophages were simultaneously exposed to either resikimod, which is toll-like receptor 7/8 agonist, or to LPS, is a toll-like receptor 4 agonist, and plasma EVs. So the secretion of IL-6 and TNF was reduced, whereas the secretion of IL-10 is this strong anti-inflammatory cytokine was augmented. So this result demonstrate that plasma EVs have an anti-inflammatory effect on macrophages exposed to microbial bumps. So to get into the mechanism behind this anti-inflammatory phenotype, we purify plasma EVs from healthy donor of both sexes using a combination of size exclusion chromatography and centrifugation, as we described in our previous work. So the isolated vesicles displayed canonical EV markers, including the tetraspanins, CD63, CD81, and CD9, and cytosolic proteins such as ALIX and HSP70. But in contrast, signals corresponding to HDL and LDL apolipoproteins, which are ApoA1 and ApoB100, were nearly absent in the EV fractions compared to whole plasma. Similarly, soluble plasma proteins such as albumin or IgG, I think it's not shown there here. Yes, IgG were either undetectable or present at very low levels, confirming the high purity of our EV preparations. Also, the transmission electron microscopy confirmed the expected morphology, while nanoflow cytometry revealed mean diameter and concentration values according to previously described work. So we have, I want to come back to this again. It's important to take a look at this. So the vitamin K dependent proteins, GAS6 and protein S, play a central role in the phagocytosis, so the process by which macrophages engulf apoptotic neutrophils. So they act as bridging factors that simultaneously recognize apoptotic cells and engage macrophages. The N-terminal GLA domains of both proteins, reaching gamma carboxylated glutamic acid residues, bind to the phosphatidylserine exposed on the surface of dying cells. And they see terminal domains in turn interact with the TAM receptor tyrosine kinases, this tyro3 AXL and MERTK, expressed by macrophages. So in this configuration, GAS6 or protein S bound to the phosphatidylserine link apoptotic cell with TAM receptors, initiating the efferocytosis and promoting downstream anti-inflammatory signaling pathways. So also, it's important to consider that the phosphatidylserine exposure is not exclusive to apoptosis. It is also observed in activated cells and in extracellular vesicles. Based on this, we hypothesize that their effect might be explained by the presence of the TAM receptor ligand GAS6 and protein S within the EV corona. So previous reports indicated that a subset of plasma extracellular vesicles expose phosphatidylserine. So we validated this finding using this single molecule localization microscopy, or SMLM, by capturing EVs either on tetraspanin-based capture, tetraspanin-based chips, or phosphatidylserine binding chips. So the bound EVs in each case were detected with fluorescent annexin 5 or with the antitetraspanin antibodies, confirming that a proportion of plasma EVs, predominantly CD9 positive, are phosphatidylserine positive. So you can see some photographs here. So each that you can see is for a single molecule. And this white circle is an estimation of the software about the extracellular vesicle surface. So continuing with this, we observed also a clear enrichment of Gas6 and protein S in EVs, suggesting their functional association with vesicles. And this was confirmed in, you can see here, it was confirmed in highly purified EVs obtained by a combination of size exclusion chromatography with iodixanol gradient or density gradient, where Gas6 and protein S were detected together with the EV markers CD63, CD9, but not with ApoA1, excluding plasma protein contamination. So complementary analysis using, again, SMLM on phosphatidylserine capturing chips and SPIRIS further show that a substantial fraction here, a substantial fraction of EVs display Gas6 and proteins on their surface. So most likely through interaction with exposed phosphatidylserine. And then because the vitamin K required for this gamma carboxylation of these GLA domains of Gas6 and protein S, a modification which is critical for the ability to bind the phosphatidylserine, we reasoned that treatment with vitamin K antagonists such as sacenocumarol will disrupt this association with EVs. Supporting this idea, EVs isolated from patients under acenocumarol therapy show a strong reduction in Gas6 and protein S levels as determined by Western blot. So all these data confirm that we can find these soluble plasma proteins, Gas6 and protein S, in the EV corona. So then, among these TAMP receptors, so MERTK is the most extensively characterized in macrophages and is known to mediate the recognition of Gas6 and protein S, thereby driving anti-inflammatory and pro-resolving responses. So consistent with this, the exposure of macrophages to EVs resulted in MERTK phosphorylation, as you can see here, at two different time points, pointing to a direct receptor engagement. A well-described pathway downstream of MERTK involves the induction of suppressor of cytokine signaling 1 and 3, SOX1 and 3, which act as negative regulators of inflammation and macrophage polarization. So in line with this, related with prosecuimod, the EV treatment significantly increased SOX1 and SOX3 transcript levels at 12 and 24 hours, as determined by qPCR. And also at the cytokine level, you can see that EVs suppressed the IL-6 expression while simultaneously promoting IL-10 transcription. So collectively, these findings indicate that EVs activate the TAMP receptor signaling in macrophages, fostering an anti-inflammatory state. Okay. And so after that, so to assess the functional contribution to MERTK, macrophages were stimulated with prosecuimod in the presence of EVs and the pharmacological inhibitors. So both the BMS 777607, a TAM inhibitor that also blocks Tyro3 and AXL, and also a selective blocking MERTK antibody. So as you can see here, first we can see the IL-6 when you simultaneously treat macrophages with EVs and prosecuimod, and then when you pre-incubate or with the anti-MERTK antibody or the BMS, this inhibition was reversed. And you can see here the isotype for control. The same with the promotion of IL-10. So when this promotion of IL-10 secretion, when you treat macrophages with EVs, when you treat, of course, activated macrophages with EVs, when you pre-treat your macrophages with blocking MERTK antibody or the BMS, you see the loss of this anti-inflammatory effect. So this indicated that the TAMP receptor signaling is critical for the modulation of to response mediated by the plasma. Finally, we use a different strategy. So we examine whether the EV-associated ligand Gas6 and PROS1 account for MERTK activation. So EVs were pre-incubated. You can see here, were pre-incubated with soluble decoy receptors, which are soluble AXL or soluble recombinant MERTK, or with also a function blocking anti-proteinase antibody before addition to the so each strategy significantly impair the ability of EVs to downregulate the IL-6 and TNF while promoting the IL-10 secretion. So these results strongly support a model in which the EV-bound Gas6 and proteinase interact with TAMP receptors, particularly MERTK, to mediate the anti-inflammatory functions.
So after having all these results and knowing that plasma EVs could do and could affect macrophages, we decided to go further in different innate immune cells and other like adaptive immune cells as well. And for that, we started by, next one, please. We started with the screening. Here in the panel A, you can see that we stained with CFCE the plasma extracellular vesicles, and we spiked different blood samples from different healthy donors, and we analyzed by flow cytometry if there was any interaction between EVs stained with CFC and the different immune cells. Basically, we analyzed PBMCs, in which we analyzed monocytes and natural killer cells, B lymphocytes and T lymphocytes, and we also analyzed neutrophils and as like the polymorphonuclear cells. And what we saw was something that is already reported in the literature, that is that monocytes interact a lot with plasma extracellular vesicles as well as macrophages, but something that was not already reported, that neutrophils were the second most abundant cells to interact with plasma extracellular vesicles, even more than any lymphocyte or natural killer. So we decided to go further in the analysis of this interaction, and we took advantage of neutrophils not expressing CD9 at all, and that recently isolated neutrophils do not expose fofadilserine in their membrane, at least not in a high amount. So we analyzed the interaction between EVs and neutrophils by these two markers, annexin 5 and CD9. So we spiked isolated neutrophils with extracellular vesicles. And after 1 hour and 6 hours, we evaluated fofadilserine and CD9. And we saw that in both time points, there was at least a 30% of neutrophils that acquired fofadilserine, annexin 5, and CD9 stain. So this suggested that they were interacting and there was no confounding factor with CFCE. Although we did all the controls, we needed to check that CFCE was not staining like by artifact the neutrophils. So by this way, we corroborated. We also corroborated by a confocal microscopy, in which we stained neutrophils membrane by CD11b, that is a canonical marker for neutrophils, DAPI for their nuclei, and for EVs, we stained them with CD9, and as you can see there, for the neutrophils that weren't exposed to extracellular vesicles, there was no CD9 stain. And for the ones that were exposed with EVs, you can see that there are a lot of green dots, suggesting also this interaction. However, we started thinking that this phenomenon could be happening in circulation because of our stringent protocol, because the isolation of neutrophils require a lot of steps of centrifugating and density gradient or sedimentation by dextran using. So that made us think that we could be losing that interaction between CD9, between EVs that expose CD9 and neutrophils. So what we decided to do and to evaluate were the same blood donor samples in a comparative way, in which we isolated neutrophils and we compared it with their whole blood neutrophils. This means that those neutrophils were isolated by gating strategy in flow cytometry and not by any isolation procedure. And we compared the CD9 percentage of both neutrophils in the same donor samples. So as you can see here, the whole blood neutrophils exposed a lot more CD9 than the isolated ones. But remember that neutrophils do not express CD9. So this suggested that circulating neutrophils present in their membrane CD9 that could be from the plasma extracellular vesicles. After corroborating that this interaction was actually happening and it could be really relevant, we decided to evaluate the different inflammatory responses of the neutrophils, as I told you before, like oxidative burst, the granulation, and the cytokine secretion. First, as you can see in the panel A, we evaluated the production of reactive oxygen species by neutrophils by using the hydrorhodamine staining, which it's a probe that when it gets oxidized, it shines, and you can detect it by flow cytometry. And as you can see here, when neutrophils were exposed to plasma extracellular vesicles and FMLP, that it's formulated peptides, and that it's not a pump, it's peptides from bacteria that activate chemioattractant receptors. You can see there that reactive oxygen species were secreted by neutrophils, but in a lower amount. As well, as you can see in the next panel, the one neutrophils that secreted reactive oxygen species, but were treated with extracellular vesicles, secreted them in a lower amount. So less neutrophils secrete reactive oxygen species or produce reactive oxygen species, and they did it in a lower amount. After that, we analyzed the granulation, and as you may know, the granules of neutrophils have some canonical markers, as such CD11B and CD66B are two canonical markers of granules of neutrophils. So we analyzed the surface expression of these two markers on neutrophils treated with FMLP or without FMLP, and in presence or absence of extracellular vesicles. And as you can see here in the violet bars, both in CD11B and CD66B, EV treatment reduced the expression of both markers in neutrophils membrane, suggesting that not only less reactive oxygen species were produced, but also that less granules were fusing with the plasma membrane. After that, we analyzed neutrophil cytokine secretion profile. We analyzed IL-1 beta, that is a canonical inflammatory cytokine. As you can see here, with EV treatment and LPS with ATP in order to the inflammasome to be really activated, secreted less amount of IL-1 beta. So following in the same direction as the previous results. However, we also saw and analyzed IL-8 and TGF-beta. For IL-8, it's a more controversial cytokine because it is always associated with an inflammatory function, but nowadays it's already known that IL-8 has a lot of functions as pro-vascular, pro-angiogenesis. So we are thinking that this increase of IL-8 after EV stimulation alongside LPS could be favoring angiogenesis. So in the line of tissue repair. And finally, we analyzed TGF-beta, that it's a canonical anti-inflammatory cytokine as well. We evaluated this by a reporter cell line, and we observed that the increase of TGF-beta dependent only of EV stimulation and not LPS, something that has already been shown that LPS do not favor TGF secretion in neutrophils, suggesting that EVs have the power to induce TGF secretion, their cells. So after that, we I'm taking into account that in circulation we saw neutrophils that had CD9 in their membrane. We started thinking that maybe there was also like EVs were inducing a phenotype or modulating neutrophils responses in circulation as well. So as you can see here in the first two panels, we didn't isolate neutrophils. We took whole blood neutrophils, as I explained before, and we analyzed their reactive oxygen species production by the dihydrorhodamine, selecting them by CD9 exposure. And what we saw was that the CD9 positive neutrophils that we selected by the gating strategy secreted or produced less amount of reactive oxygen species after FMLP stimulation, as you can see in the following panel, suggesting that this interaction that occurs in circulation also favors neutrophil decrease of inflammation response. On top of that, we determined extracellular vesicles function in an in vivo model of mice. We induced peritonitis by chymosan stimulation in the peritoneal cavity, simultaneously with PBS or plasma extracellular vesicles in four mice at each group. After 18 hours, we did a peritoneal lavage. With that lavage, we took all the cells that were recruited in the peritoneal, and we analyzed them by flow cytometry. I am not showing you here, but we didn't really we couldn't really distinguish a different number of recruited cells, so there was no difference there. But when we stimulated with FMLP and evaluated neutrophils reactive oxygen species production by dihydrorhodamine, we observed that those cells that were evenly recruited in both type of mice, the ones that have been exposed to extracellular vesicles secreted less amount of reactive oxygen species, suggesting that EVs could be influencing, suggesting the plasma extracellular vesicles were inducing an anti-inflammatory phenotype on neutrophils, decreasing their reactive oxygen production. There were less neutrophils producing them, and in a lower amount as well. So validating our model in vitro and in an in vivo model. So we started thinking about the possible mechanisms that could be being involved in these mechanisms by which EVs are modifying neutrophils. And we started thinking about adenosine, that is a final product of ATP cleavage. As you may know, ATP is an inflammatory molecule that it's rapidly cleavage by sequentially by two enzymes, mainly that it's CD39 that cleaves ATP and ADP into MAP, and CD73 that is the enzyme that is responsible for cleaving MAP to adenosine. Adenosine has several inhibitory or anti-inflammatory functions, and it is mainly by activating A2A receptors. in neutrophils. So we started thinking that maybe in our system, in somewhere, adenosine was being produced. So we started thinking that maybe EVs were carrying these enzymes. After some biography, the previous one, please. So we ran into this paper that describes a similar approach that we were thinking, in which EVs were carrying in their membrane, as part of their membrane, CD73 as an active enzyme. So after that, we wanted to corroborate that our EVs could be carrying the enzymes and not as part as soluble proteins. So the next one, please. So first of all, we evaluated our preparations by Western blot. As you can see here, the EV fractions that were pulled from after SEC were evaluated for CD73 and CD39 in 3 different donors or 4. And they were also associated with canonical markers of EVs that, for example, CD9. In the panel B, as you can see, we showed that they were in the same fractions that EVs, confirming this previous result. So these enzymes were carried by EVs. After that, we corroborated these results by 2 single EV approaches, by nanoflow cytometry and single molecule localization microscopy, in which both techniques showed us similar results, in which there were 4 different subtypes of EVs. The majority were negative for CD39 and CD73. However, there were 3 populations that were positive. 10% of them were positive for each of the enzymes, single positive, and 1 or 3% were double positive for the enzymes, which takes into around 25 or 20% of the EVs carrying these enzymes. So after that, what we decided to do, or what we aimed to do, was to corroborate the next one, please. If those enzymes that were carried by EVs were active. So first of all, we evaluated CD39, as you can see in the right panels, by a luciferase assay, which consumes ATP and functions as a luminemetric assay. We showed that our preparations were active for ATP cleavage. We could determine the ATPase activity by different doses of EVs. And in the panels C and D, we could also determine the ATPase activity by exposing the assay, the EVs, to different concentrations of ATP. So so we concluded that CD39 activity was there and EVs hydrolyzed ATP. Then in a similar approach as luminemetric assay, but it's different, it doesn't take into account luciferin and luciferase. What we did was evaluate CD73 activity. What we observed was that after 30 minutes and 60 minutes, the amount of MAP decreased, so suggesting that CD73 in plasma EVs were active as well. However, this didn't mean that these enzymes present in the EVs were the main mechanism, or were involved in the mechanism by which EVs reduced oxidative burst and degranulation. So we decided to test that. The following, please. In order to test that, we exposed neutrophils to 3 different compounds. As you may see here in the left panel, we treated neutrophils with POM1, that is an inhibitor of CD39. Then ZM, that is an inhibitor of the main receptor that I told you before, A2A, and the ADA, that it's the adenosine deaminase. As you can see here, adenosine, that is the last product of ATP cleavage, can be further cleaved by this enzyme into inosine, that is a compound that has not any anti-inflammatory immune function. So we treated neutrophils with extracellular vesicles with the different compounds, and as you can see here, we analyzed the reactive oxygen species production by dihydrorhodamine as well. And as you can see here, the treatment with different doses of the POM1 of the inhibitor of CD39, ADA, that is adenosine deaminase, and ZM reverted the effect of EVs, suggesting that this is the main mechanism by which EVs regulate neutrophils' oxidative burst. As a positive control, you may see here in the right panel, we used adenosine by itself in order to corroborate that it could modulate neutrophils' reactive oxygen species production, and it was also reversed. So in order to conclude this part, we demonstrated this way that adenosine and both enzymes CD39 and CD73 are key for regulating neutrophils' immune response. Now, the following. So after all these results, we started thinking that maybe extracellular vesicles that were attached or interacting with neutrophils that expose phosphatidylserine in their membrane and the bridging molecules, as we described before, could be favoring macrophages to phagocytose those neutrophils, as recognizing them as apoptotic cells without being apoptotic. So we designed an aphagocytosis assay in which we co-incubated macrophages with the EV-treated neutrophils, and we analyzed their interaction and if there were any phagocytosis. We did it by flow cytometry in panels A and B, in which we stained neutrophils with CFCE. And as you may see here in the green panel, there is a positive control done with apoptotic neutrophils, in which you can see this population after the main one, that is between 10 to the 3 and 10 to the 4th, and that means there is aphagocytosis. The macrophages are phagocytosing neutrophils, apoptotic neutrophils. As you can see in the following 2 panels, where it is neutrophils treated, living neutrophils treated with EVs and in presence or not with LPS, you can see that that population does not appear, suggesting that macrophages are not eating living neutrophils. However, a second population appears that it's high in CFC stain. This could suggest that macrophages were interacted with neutrophils stained with CFC, because the phagocytosing process eliminates a little bit of the CFC stain, so you can see it in a lower MFI, and here we can see it in a higher amount. So we counted the percentages of these interactions that we call duplets, and we could see that EVs favored these duplets and these interactions between macrophages and neutrophils. So we tried to confirm this result with a confocal microscopy, in which we stain macrophages that are really rich in CD14 with CD14-50. Neutrophils were stained with far red, and they were stained both with DAPI for their nuclei. And as you see here, EV-treated neutrophils interacted more with macrophages than the ones that were not treated with EVs, confirming that this interaction was happening. However, we didn't know if this interaction could be modulating macrophages' response. So we designed an assay in which, instead of flow cytometry or microscopy, we analyzed the cytokine production of macrophages upon neutrophil co-incubation. So we co-incubated EV-treated or not neutrophils with macrophages in presence or absence of LPS. And what we saw was a similar phenotype as the macrophages we explained before. We saw a decrease on IL-6 secretion and IL-10 increment in the production of IL-10 incremented, suggesting that there was a M2-like phenotype or more anti-inflammatory phenotype of macrophages. Taking into account the previous results, we started thinking that maybe the Gas6 and protein S attached to the EV could be the mechanism by which this interaction was happening. So we treated macrophages, pre-treated macrophages with the anti-MERTK blocking antibody and the BMS, the same as Alan mentioned before. And what we saw there when we incubated neutrophils treated or not with EVs was that decrease of IL-6 and the increment of IL-10 was reversed, indicating that Gas6 and protein S were the mechanism involved in MERTK engagement and the downstream anti-inflammatory pathway. So what I want to say is that these results are really important, as this could be until now an unknown mechanism by which neutrophils can modulate macrophages without even being apoptotic. So starting an anti-inflammatory pathway before and faster than being apoptotic. So this was really cool to be discovering it. Now the following. So in order to conclude, what we have been talking about this time is that plasma extracellular vesicles expose phosphatidylserine, and by bind Gas6 and protein S. The important thing here is that it was already known that phosphatidylserine was exposed, but we, by SMLM, we discovered that there was a lot of EVs that expose phosphatidylserine. Then that these bound Gas6 and protein S are able to engage MERTK receptor, modulating macrophages and favoring an M2-like phenotype and anti-inflammatory phenotype, reducing IL-6 and increasing IL-10, as well as reducing TNF. Also that plasma extracellular vesicles modulate neutrophil activation, principally by the sequential cleavage of ATP by CD39 and CD73 in EV's membrane, and the finally formation of adenosine, by which it acting in A2A receptor. Overall, we propose that plasma extracellular vesicles are endogenous homeostatic modulators. So that's it for now. We want to say thank you to all the people involved in the project, and our collaborators, and every funding organization as well.
All right. Well, thank you so much, Martina and Alan, for sharing your work here. We do have a few questions in the chat box, and I know we're coming towards the end of the hour. But if anybody else has questions, please do put them in the chat box, and I'll call on you. So, in the interest of time, though, I think I'm going to just go through the questions that we already have. So, Bhuvan asks, did you also check the role of EVs on neutrophil functions? I think you showed some of that. But maybe you just want to respond to that question.
Yes, well, we showed different functions. Reactive oxygen species production, that it's key for phagosome killing of pathogens. Also the granulation that helps with pathogen as well. Obviously, if it is not controlled, it can lead to tissue damage. So it's good that EVs can modulate it. And we also checked well, we didn't show it here, but we checked metalloproteinase activity, and it was also decreased. Metalloproteinase 9 is also in granules. So by other technique, we also were able to corroborate that. Yes.
And Bhuvan is also
cytokine
asking,
profile.
Bhuvan is also asking about the concentration, so a dose-dependent effect of EVs on MERTK activation, which I believe you showed, but maybe you just want to say a couple of words about how you decided what doses to use.
So let me get to the here this phosphorylation of MERTK, the question.
That's right.
So we decided, so we showed here the EV med medium or EV high dose, but also we have a EV low dose. So we decided by also previous work that is here. And so regarding the phosphorylation of MERTK, we didn't see a dose dependence effect on the MERTK phosphorylation. So we did so on a time dependent mechanism. Yes, and regarding the exactly concentrations, we can check here. So the EVs isolated from 2 milliliters of plasma were concentrated in 60 microliters of PBS, and then we decided the doses. So this 1/4 is for the low dose, and this 3/4 is the high dose. So this is the approximation of the concentration. If you take the numbers of the nanoflow cytometry, you can exactly calculate the number of EVs that we use in each experiment.
Good. Okay, so hopefully that answers Bhuvan's questions. And then Phil Askanase introduces two interesting concepts here in the chat box. One of those is about the possibility that when you treat cells with EVs, you actually have EVs that are then produced by the treated cells that could have similar effects or even opposite effects. So I'm wondering if you want to speculate on what you think might be happening in these cultures in terms of the EVs that are being released by the cells that you treated with your EVs.
Well, sorry,
No, go ahead.
If I can go, okay. Regarding neutrophils, extracellular vesicles, there is a lot of studies that show that it depends on the stimuli that they receive, the kind of EVs that they secrete, something like tailored. So I would speculate that neutrophils are also secreting anti-inflammatory extracellular vesicles. We have not studied them, but it could be.
Regarding
Yes.
macrophages, the macrophages, they don't release too much. They don't release a high number of EVs. So in comparison with plasma, when you have millions and millions, but there are some work in mostly mice that demonstrate that an M2 macrophage or M2-like macrophage has that the EVs isolated from M2 macrophages have anti-inflammatory activity in the context of sepsis or SIRS or some systemic inflammatory disease. So we can speculate. I can speculate the same that Martina said.
Yeah, and indeed, Phil does mention the M2 like macrophages here. The final thing, and I'll let you go. I'll let you all go soon. But the final thing that Phil introduces here is the concept of the PMN nuclear traps, right? So these neutrophil traps that are released. What do you think might be happening there with EVs? Are EVs contributing to that? Do the traps prevent access to cells by EVs? Have you thought about those traps at all?
We tested nets on we tested extracellular DNA after EV treatment of neutrophils, and we could see that there was less amount of extracellular DNA. However, nets need to be characterized and studied in multiple ways because it is otherwise not complete and not for sure. So we have this approximation that EVs really, plasma EVs decrease, could decrease net formation. And obviously nets can, maybe they can limit the access of plasma EVs to inflamed tissues, or maybe they could favor the phagocytosis by macrophages. But I really don't know. What I do know is that there could be something on net release and EV stimulation.
Great. All right. Well, we will conclude then for today, but thank you so much again, Martina and Alan and Matthias, for leading this project. And thanks to everybody who joined today and for the great questions and comments. I look forward to seeing you all again at a future EV Club and wish you all the best with your research. Take care, everybody.
Thank you very much.